For having me here. The morning session was fabulous. It was great to see how Friends of Cancer Research really sort of convenes and catalyzes all these stakeholders together. And I like that term earlier about providing freedom to operate to answer critical policy questions. So Jeff wanted me to discuss some data that we presented at ASCO this year. And it was really looking at our legacy clinical trial data sets in non-small cell lung cancer submitted as NDAs or BLAs, and looking at a very sort of pragmatic, real-world endpoint, time to treatment discontinuation. It's very, in contrast to some of the other endpoints, like overall survival, progression-free survival, response rates, it's pretty simple. You know, when did the patient start the treatment? When did they stop the treatment? So very easy to understand, and it's sort of a composite of both safety and efficacy, if you will, because you know, patients who tolerate the drugs for a long time are able to stay on drugs longer, and patients who benefit from the drugs are able to stay on drug longer. And in contrast, patients who don't tolerate the drug or don't derive benefit come off quicker. So just a little bit of background. As we all know, progression-free survival, or PFS, is an important efficacy endpoint frequently used in oncology clinical trials. Increasingly, as was alluded to this morning, trials and doctors want to keep patients on beyond objective radiographic progression, so-called resist progressive disease, for patients who are deriving benefit. This is for a few reasons. One, drugs are a lot better tolerated these days. So we have targeted therapies, immunotherapies, better tolerated than chemotherapy. And for patients without good treatment options, you know, patients and doctors want to keep going. The other thing to mention is resist progression is sort of an arbitrary cut point. You know, this 20% growth in some of target lesions or new lesions. This is, you know, it's standardized, so we have benchmarks to compare with historic trials, but it's an arbitrary definition which doesn't necessarily correlate with disease biology. So particularly for immuno-oncology agents, where you might get a growth on scan, which might be T-cell infiltration, or targeted therapy drugs, where you might get a growth of an existing lesion or even a new lesion. Patients might still derive benefits by suppressing that driver oncogene beyond traditional resist progression. And as we have heard this morning, time to treatment discontinuation has been proposed as a potential real-world endpoint. It can be easily abstracted from the EMR. As we heard this morning, progression-free survival is a lot harder to derive in the real world. Assessment periods aren't uniform. How to define progression is challenging out of EMR data. So we asked two key questions. Number one is, how well does time to treatment discontinuation correlate at the patient level in recent lung cancer trials? We divided it by different types of trials. We had ALK positive patients receiving tyrosine kinase inhibitors, or TKIs. We had EGFR mutation positive patients receiving TKI. We had sort of EGFR wild type or unselected patients receiving TKI. We had patients receiving platinum doublet chemotherapy, and then patients receiving docetaxel monotherapy. And then our second question was related to outliers. How many outliers exist in these legacy clinical trial data sets? We defined sort of late TTD as patients who stayed on therapy for at least 3 months beyond traditional progression. And then early TTD, in contrast, are those who discontinued therapy at least 3 months before the progression date. And we collected and analyzed data from 18 randomized controlled trials in lung cancer from 2007 to 2014. So this is a high-level summary of the results. I realize it's very busy and maybe not conducive to a deep data dive. But I just wanted to call attention to a few points. If you look at that top row, those are all the patients. So we had about almost 9,000 patients. And you can see that the patient-level correlation between TTD and PFS was fairly robust at 0.87. And you can also look at the median PFS and median TTD across these different patient cohorts. And interestingly, for example, for the EGFR mutation patients, the median TTD exceeded that of the median PFS, same with ALK. Whereas in contrast for immune checkpoint inhibitors and chemo, it was the contrast. So the median PFS was longer than the median TTD. The other thing I would mention is for immune checkpoint inhibitors, these approvals were based mainly on survival benefits. If you looked at many of these trials, particularly immune checkpoint inhibitor versus docetaxel, there was no difference really at the medians between trial arms. So this is just a sample anonymized Kaplan-Meier curve. This is a trial of EGFR tyrosine kinase inhibitor versus chemotherapy in EGFR mutant patients. And the blue line is the light blue line on top is the TTD and the EGFR TKI arm. The pink line is the PFS. And then you can see those two bottom lines are the chemo, the yellow and the darker blue. And you can see that in this trial, at least, TTD and PFS seem to track similarly. I want to show this example of a trial of also EGFR TKI versus chemo, but here there was no maintenance chemotherapy given. So there was a steep drop-off for patients who were given doublet chemotherapy, which just goes to show you this endpoint is not great if there's a scheduled discontinuation of treatment, and probably should not be used if you're only giving, say, 4 to 6 cycles of doublet chemotherapy and then stopping, or 4 cycles of ipilimumab and then stopping. Through the help of Yutao Gong, who's here today, we also did some scatter plots looking at sort of patient-level correlations between TTD and PFS. The bright dots are patients who have had events for both TTD and PFS. The gray are those who have been censored for one or the other or both. And you can see, again, sort of in the EGFR mutant population, there seems to be pretty good correlation between TTD and PFS. Immune checkpoint inhibitors, there appears to be more scatter, in both directions, actually. And then with doublet chemotherapy, there also appears to be more scatter. More patients who had long progression-free intervals but were taken off therapy, possibly due to toxicity. And then we also looked at these histograms. We looked at outlier effects. And what you can see here is that EGFR mutant patients and ALK patients had a lot of late TTD, so 12% EGFR mutants, 23% of patients with ALK rearrangements had a TTD minus PFS of at least 3 months. With immune checkpoint inhibitors, it went both ways. There was some late, there was some early. And with platinum doublet chemotherapy, a lot of patients came off treatment before their defined progression date. So what can we conclude from all of this? Apparently there's a strong patient-level correlation between TTD and PFS with an overall correlation of 0.87. The correlation may be stronger for daily administered tyrosine kinase inhibitors than those IV infusions, which are given every 2 or 3 weeks. We saw that the median time to treatment discontinuation was greater than the median PFS, particularly for oncogene-directed tyrosine kinase inhibitors. And then the reverse was true for immune checkpoint inhibitors and chemotherapy. And again, we saw this meaningful minority of outliers with late TTD, with ALK patients, EGFR patients, some immune checkpoint inhibitor patients, and then early TTD with chemo and immune checkpoint inhibitors. We have Raji and the statistical group in the room, so I'd be remiss not to point out some limitations of our study. This was retrospective. It was post hoc. There were differential censoring patterns between PFS and TTD. There's a lot of heterogeneity. Keep in mind these trials range from starting in 2007 to 2014. The sort of permissiveness of regulators and trialists and investigators in allowing treatment beyond progression sort of progressed over time. So we see more allowance of treatment beyond progression in more contemporary clinical trials.